All of the details of everything that I'm going to talk about, the papers, the data sets, everything else, the software, everything is here at this website. And then here are some more personal thoughts about what some of it means. So what I'd like to do today is go over 3 main points. The first is that I'm going to talk about problem solving in unconventional substrates, and specifically this idea of living tissue as an agential material. And this is part of the bigger field of diverse intelligence, and I'll talk about that. I will use one specific model system as an example of what I call cognitive glue, which is policies among components that solve the scaling problem of having an emergent cognitive system out of smaller parts. And we will use biological morphogenesis as a model system to think about developing tools and strategies for analyzing these collective intelligence systems. And at the end, I want to show you some novel living beings that may help us to think about the origin of both spatial and cognitive patterns and where they might come from. And basically what my group does boil down to one sentence is that we study collective intelligence, for example, how groups of cells navigate the space of anatomical possibilities, and we try to push some pretty philosophical ideas about that into actual therapeutics. And so for us, this is how we test whether these ideas are useful or not. So let's start by thinking about this kind of thing. You know, this is a very old classic painting. Adam is naming the animals in the Garden of Eden, and the worldview that is shown here is that basically there are some discrete natural kinds, and so here's Adam, here are these other animals, and we can study the various properties that these other creatures have. But it's nice and easy because they are all discrete and different from each other. It's very easy to say who's who. So that's you know, even though it's a really hard task, of course, to understand the cognition of other species, we can sort of call it the easy problem in Chalmers' sense, because at least we know what everything is here. But the problem is that actually these discrete kinds don't really exist, because all of us are at the center of several different continuous lines of possible beings. So both at the level of individual development and of course evolution, we are just one point out of a very slowly changing set of diverse forms. And whatever you want to say about this modern human, you have to ask yourself, well, at what point and how did these capacities come to be? And then because of approaches developed in bioengineering and genetics and synthetic morphology, we now know that both biologically and technologically, we are in the middle of a very large continuum of possible hybrid beings, because at any point we can introduce into any level of organization, we can introduce synthetic and engineered materials, and life is incredibly interoperable, and so you can make these things, and of course people are and will be increasingly doing that. And so again, whatever philosophical or conceptual framework we have has to be able to handle all of this, not just the difference between specific distinct discrete species, but be able to say something about all of these different kinds of possible variants. So what my framework tries to do is to help us to recognize, create, and ethically relate to truly diverse intelligences. So regardless of their composition or how they came to be, I'm interested in, of course, familiar creatures such as us and other primates and birds and so on, but also unusual colonial organisms and swarms and synthetic biology, so engineered new life forms, AI, whether purely software or embodied in some kind of robotics, and someday maybe exobiological agents. And so what I'm interested in are ways to think about all of these things in a way that moves experimental work forward. So it gives us new capabilities, new discoveries, and hopefully better ethical frameworks. And so all of that is detailed here. But of course, I'm not the first person to try for this. So here's Rosenbluth, Wiener, and Bigelow's attempt to make a scale going all the way from basically passive matter all the way up to this kind of second order metacognition that humans have. And so this is trying to figure out what that continuum looks like has been an interesting problem for a long time. So the way I try to think about it is through what I call an axis of persuadability. Basically, what I try to understand is what kind of tools are we going to use for a given system to interact with it, basically. And the idea is that this is kind of an engineering approach, because then it becomes an empirical question as to, for any given system, where along the spectrum does it lie, and cognitive claims become interaction protocol claims. So, for example, for a given system, if you say it has this or that level of intelligence or cognition or whatever, what you're really saying is, how can we interact with it? Do we just modify the hardware, or can we reset some set points? Can we train it? Does it respond to logical arguments, and so on? There are lots of different sciences, from control theory and cybernetics and behavior science and psychoanalysis and so on. And so the idea is that for any given system, you really can't just assume where it is. You can't have philosophical feelings about where things should be. You have to do experiments. You have to ask. And it turns out that we get a lot of surprises now. And so one of the things that we're interested in is intelligence in novel spaces. So we, as humans and as scientists, are reasonably proficient at recognizing intelligence in familiar three-dimensional space. So medium-sized objects moving at medium speeds, birds and primates and so on, maybe an octopus or a whale, and we can kind of see them doing interesting, intelligent things. But biology does the exact same thing in many other spaces. So our familiar 3D space is not the only space where intelligence is deployed. So there is the space of possible gene expression. It's a very high dimensional space. There's a space of physiological states. And what we will talk about today is this anatomical morphospace, the space of all possible configurations of a body. And within any of these spaces, even though it's difficult for us to visualize, if you use the tools of behavior science and cybernetics and some other things, you can actually observe and make use of the fact that these systems have intelligence in navigating those spaces. So let's talk about this one in particular. We could do many hours on these other ones, but let's just focus on morphospace. We have to understand that our intelligence at one point, both evolutionarily and developmentally, was a little blob of chemistry and physics. So this is an unfertilized oocyte, and it's slowly, gradually, it becomes something like this, or even something like this. And we all make the journey from something that looks like it's just chemistry and physics to something that's obviously has a mind and various other cognitive properties. So what we need to do is figure out how we got from here to here. What actually happened during this journey? So it's a little weird for many people to focus on development and realize that basically at one point they were just an unfertilized oocyte. But at least we are a true centralized intelligence, right? We have this nice brain, and we're not like these other supposed intelligences that people call ants and termites and bird flocks, collective intelligences. But many people think that that's not real intelligence the way we have it. We have a nice centralized brain, and in particular, Rene Descartes really liked the pineal gland in the brain because it was an unpaired structure, and he felt that it was appropriate for the kind of unified cognition that humans enjoy. But if he had good microscopy, he would have looked inside this pineal gland, and he would have seen that actually there's not one of anything anywhere, where basically inside that pineal gland is a very large number of cells. Inside of each of those cells is all of this stuff. And so in the end, it turns out that we are all collective intelligences. We are all made of parts. And the real trick is to tell some kind of useful story about scaling, how it is that the properties of the parts give rise to the cognitive properties of the whole. And this is the kind of thing we're made of. This is a single cell. This is called the lacrimaria. There's no brain. There's no nervous system. You can see it carrying out its daily activities here. It's hunting for food, and all of that is handled within the single cell. So we are made of a kind of very, very competent, very active material. We're not made of passive materials the way that our current robotics and other types of engineering. Sometimes I give a talk called "Why Robots Don't Get Cancer," and this is why. It's because they are not made of this multi-scale kind of material that has agendas. In fact, one thing that we can now see is that even within a single cell, never mind all the structures of the cell, but just the molecular pathways, in fact, very simple, even very simple molecular pathways, have learning capacity. You can train them. If you start to stimulate and record various components in a gene regulatory network, you can see that they're capable of at least six different kinds of learning, including Pavlovian conditioning. And so all of that is described here. And we are at the moment now trying to take advantage. So we're building devices to train these cells, and it has lots of applications in medicine, for example, for drug conditioning and things like that. So even inside of our individual cells, you already have the basic components of intelligence and various other behavioral properties. So what we're looking at in our bodies is this multiscale competency architecture. That is, at every level, we've got components that are solving problems in their own space. So there are different spaces, and all of these components, the molecular networks, the cells, the tissues, the organs, and so on, are competent to do various things. And so let's then ask what are the competencies of cells in morphogenesis? How do they create our amazing complex anatomies? So this is a cross section through a human torso. You can see there's an incredible complexity here of all the different organs in the right place, the right size, the right shape, next to each other. But we all started like this, a little ball of blastomeres. And so the question is, where did this come from? How did these cells build this? And you might be tempted to say, well, it's the DNA, the genome explains this. But of course, we know that the genome doesn't directly specify any of this. The genome specifies proteins. And that protein hardware that's available to every cell is running a kind of physiological software that enables these cells to know what to build and when to stop. And so we need to understand how that works. We need to understand how to enforce changes when something is damaged or missing. How do we get the cells to rebuild it? And as engineers, we can also ask, well, what other shapes are possible? This hardware, what else can it build? And it's important to make analogies to kind of to the facts of behavioral science, where we know that the structure of the termite colony or the precise structure of a spider web are not directly specified in their genome. What the genome gives you is a very competent agent that is able to then through the physiological workings out of what the cells are capable of making these other patterns. So the way that we can formalize this problem definitively is to think about the end game of regenerative medicine. What is it that we want in the end? Well, in the end, we want something like this. I call this an anatomical compiler. So the idea is that someday you will be able to sit in front of a system like this, and you will be able to draw the plant, animal, organ, or biobot that you want. In this case, we've drawn a nice three-headed flatworm here. And the idea is that if we had this anatomical compiler, what it would do is take this shape and compile it into a set of stimuli that would have to be given to cells to get them to build exactly this, right? So the idea is, if we knew what we were doing, we would have complete control over growth and form, and you would know what stimuli should you give to cells to get them to build anything, whatever you wanted. The reason we need this is pretty obvious. Basically, if we had something like that, then birth defects, traumatic injury, cancer, aging, degenerative disease, all of this would go away, because we could tell the cells what new tissues to build. The reason we don't have anything like this, even remotely, and despite the fact that molecular biology and genetics have been making great progress over many decades, is that we've been thinking exclusively at the level of hardware. And what I think we need to realize is that this system is not something like a 3D printer that micromanages the position and the gene expression of the cells. It's a communications device. What you're trying to do is translate your goals to the goals of the cellular collective, and that requires thinking about this in a very different way. So in modern molecular medicine, we're very good at the hardware. We're very good at figuring out which genes interact with which other genes and proteins and so on. We're actually really far away from large-scale control of form and function. If somebody's missing a limb or a birth defect or a tumor, we're still really very primitive in all of this. And I think that's because we've been assuming that the biological material is merely complex. And it is complex, but that is only the beginning of the story. It's not just complex, it's actually an agential material that has its own competencies and agendas. And I think regenerative medicine is going to be completely transformed when we adopt some tools from the cognitive and behavioral sciences to deal with this material. I've used this word now a bunch of times, so let me try to define it for the purpose of today's talk. I do not claim that this is the one definition that subsumes all of intelligence. This is just one slice of it that I want to talk about today. So I like William James's definition, which is that it's some degree of ability to reach the same goal by different means. So the idea is that when you want to understand the intelligence of a system, you have to hypothesize a problem space that it works in. You have to hypothesize what the goals are. You can hypothesize what competencies it might have to reach those goals despite various interventions. And then you do the experiment and you see how well that worked out for you. So we can test it in very simple systems like two magnets separated by a barrier. They're very stupid. They will never go around the barrier to meet each other because that requires them to temporarily get further from their goal. So all they can do is go down a smooth gradient. Here Romeo and Juliet, in contrast, they have all kinds of skills and planning and all kinds of memory, all kinds of ways to avoid and go around social and physical barriers. And then in between, you have all sorts of things. You have autonomous vehicles, you have cells and animals and so on that have some degree of navigation ability. So this is what I'm talking about when I say intelligence. I mean specifically navigating a problem space with novel barriers to get your goals met. Okay, so now let's think. What kind of collective intelligence do cellular swarms have? Well, we know that development is incredibly reliable. So almost all of the time, a few embryonic cells will give rise to a normal human anatomy. And we also know that this process involves a huge increase in complexity. But that is not why I'm calling it intelligence. Intelligence is not simply being reliable or simply generating complexity. Those are very easy to do, and there are lots of very simple systems that do that. This is not about this is not about just complexity and reliability. It's really the problem-solving capacity. And you start to see that when you start to perturb the process, not merely observe what it does normally, but you start to perturb it. So for example, what happens if I cut this early embryo in half? So now half of this thing is missing. We know what happens if you cut any of our machines in half. What happens if you cut this? Well, you don't get two half bodies. You get two perfectly normal monozygotic twins, because this system can start from many different starting configurations. In fact, you can scramble these cells up to a certain point. You can take cells away, you can add cells. There's all kinds of things you can do, and they will navigate this space. They will go around local minima and maxima in that space, and they will reach this ensemble of goal states that represents the normal human target morphology. So the system can tell that half of it's missing. It knows what it needs to do to fix it, and eventually gets where it's going. Some animals can do this throughout their lifespan. So this is an axolotl. These guys regenerate their eyes, their limbs, their jaws, portions of the heart and brain. And for example, here in this limb, if it's amputated at any particular point here, the cells will very rapidly grow and regenerate exactly what's needed, and then they stop. And that's the most amazing thing about regeneration, is that it knows when to stop. How does it know when to stop? It stops when the correct salamander arm has been completed. So this is really an example of a kind of anatomical homeostasis. The system is quiescent here, and when it's deviated from its correct position in anatomical space to here, it will work really hard to get back. And when it gets back there, it stops. By the way, this is not just something for worms and salamanders. Humans have it somewhat, and other mammals. So we regenerate our livers. Deer regenerate massive amounts of bone and vasculature and innervation. They grow about 1.5 centimeters per day when they're regrowing their antlers. And even human children can regenerate their fingertips below a certain age. So this is a kind of a generic capacity that's really widespread throughout the tree of life. And a simple version of that kind of homeostasis can be shown here. This is a tadpole. So here are the eyes, here's the brain, here's the gut, here are the nostrils. And this tadpole needs to become a frog. In order for a tadpole to become a frog, their face has to rearrange. The jaws have to move forward, the eyes have to move, everything has to move. And it used to be thought that this was a hardwired process. I mean, every tadpole looks the same, every frog looks the same. So if you just move all the pieces in the right direction, the right amount, you will get a normal frog. So you would think you could do this with a very hardwired sort of fixed process. So Laura Vandenberg in my lab decided to check that. And again, to really understand the intelligence of a system, you can't just observe it, you have to do perturbative experiments. So what she did was she made these Picasso-like frogs. Basically, everything is scrambled. The eyes on the back of the head, the mouth is off to the side, everything is scrambled. And what she found is that these guys make largely pretty normal frogs because all of these organs move in unnatural, novel paths. Sometimes they even go too far and they have to come back a little bit, but they keep rearranging until you get a normal frog face. So what the genetics gives you is not a hardwired set of movements. It specifies a problem-solving system that can recognize unexpected states. It has some sort of ability to get to this goal state, and it will do what it needs to do to get there. So that raises an obvious question. How does it know where it's going? How does it know what the correct frog face is? And so that means that we have added to the standard. So this is the standard story that you'll get from a developmental biology textbook. It's a kind of feed forward open loop process. There's gene regulatory networks. Some of those genes become effector proteins. They do things. They have physical properties. They're sticky, or they move, or they apply force or something. And then eventually, all of that complex stuff together gives you this kind of emergent thing. So that's very much the mainstream view of developmental biology, is that it's mostly a kind of emergent complexity that happens from low level rules acting in parallel in large numbers. Well, as I've just shown you, there are some amazing examples, and I could show you many more amazing examples of a kind of anatomical homeostasis here, where if you're deviated from this final goal by injury, by mutations, by teratogens, many different things, if you're deviated, actually some feedback processes will kick in, both at the level of genetics and physics, to get you back here to reduce error. It's an error minimization scheme. And that's quite different from systems that are feed forward, like cellular automata and things like that, which typically do not have any kind of even primitive cybernetic features like this, where they try to maintain specific states. The foundation of goal-directed activity here. So what we've tried to do is to follow this kind of scheme experimentally, and to ask if this is true, where is the set point encoded? Could we learn to, well, could we find it, first of all? Second, could we read it and interpret it? And most importantly, could we change it? Because the amazing thing about these kinds of processes, like the thermostat in your home, is that in order to change what it does, you do not have to physically rewire the system. So the current commitment of molecular medicine, that you have to act down here, you have to change genes and molecular networks, CRISPR, those things, and then hope that the effects percolate properly to the system property that you're actually trying to control. If this is correct, you don't need to do that. You can actually control the set point and then leave it alone and let the system do what it does best. So this is the research program that we started, and in particular, I want to generalize it a little bit by pointing out here the similarities to neuroscience. So if you train a rat to press a lever and get a delicious reward, there is no individual cell in this rat that has that associative memory. So the cells at the bottom of the feet interact with the lever. The cells in the gut get the delicious reward. Who owns the associative memory here? Well, the associative memory is owned by the rat, which is a collective that's held together. So all the neurons in the other cells are held together by a kind of cognitive glue. Now, which solves the scaling problem. It allows the collective to have memories that do not belong to any of the individual subunits. It's a distributed kind of system, and we know how this works in neuroscience. It's implemented by a bioelectric, in this case, neural network. So what we have here is that the hardware looks like this. These are neurons that have ways of setting voltage potential via their ion channels. They may propagate states and potential to their neighbors through electrical and other kinds of synapses. So we'll look at mostly these gap junctions. And it is kind of a commitment of neuroscience in general that if we were to read the electrical activity, the physiological software that actually is the activity of this electrical network, we should be able to extract the memories, the goals, the preferences, right? So the commitment here is that all of the cognitive features of this animal are in some way encoded in the electrophysiological signaling between these cells. And this is a video that this group made of a living zebrafish brain in action. So that's the idea. Well, it turns out that this amazing architecture is very ancient. Evolution noticed that bioelectricity was good for solving problems and aligning components within collectives long before nerves appeared. It actually showed up around the time of bacterial biofilms. And it turns out that every cell in your body has ion channels. Most of them are coupled into electrical networks with these gap junctions, and most of the same molecular mechanisms, and it turns out many of the algorithms are actually quite similar between what's going on in the nervous system and what's going on in the rest of your tissues. So then we can ask the question, before nerve and muscle came on the scene and were used to think about navigating the body through three-dimensional space of behavior, what did these networks think about? What were they doing? And we can also think about a similar neural decoding kind of project, except not in neurons. So to ask here, so this is again this idea of reading the electrophysiology of these cells, decoding them using various algorithms, and could we learn what the information content really is? So this is a video of an early frog embryo using a voltage-sensitive fluorescent dye. This was made by Danny Adams in our group, and you can see all the electrical conversations that these cells are having with each other long before there's any nerve or anything like that. So what are they doing? Well, in order to find out, we developed the first molecular tools to read and write non-neural bioelectrical information. So this means voltage-sensitive fluorescent dyes that we adopted to non-invasively read these states, and now there's also genetically encoded reporters. Then we do a lot of computer simulations to understand, to link the expression of ion channel genes to the capabilities of the excitable medium that they underlie, so the electrical propagation through the tissue and all the states, and how they do things like pattern completion, which are important for regeneration, of course. I want to show you an example of what these bioelectric patterns look like. Here is this is also the work of Danny Adams. This is we call this the electric face. So this is an early frog embryo putting its face together, and you can see there's a lot going on. But if you freeze one of the frames, what you see is that here, long before the actual face gets created, already this is we already see what it's going to be. This is where the animal's right eye is going to be. This is the mouth. There are some placodes out here. The left eye comes in slightly later than that. And so what you can see is that this pre-pattern, we're literally able to read the set point, the homeostatic set point towards which these cells are going to work. And if as I'll show you momentarily, if you change that set point, then everything changes. The gene expression changes, the craniofacial patterning changes. So this is the normal endogenous pattern memory that is required to make a correct frog face. This is a pathological one. What we've done here is inject a human oncogene into these cells. These cells are going to basically disconnect from the electrical network and go back to a very amoeba-like lifestyle where the rest of the body is just external environment to them. But you can catch this happening very early on, and this is part of our cancer program that I'll mention briefly. That you can immediately see where this is going to happen. Okay, so reading these patterns is important, but again, the most important thing is functional perturbations. What do these patterns do? Are they actually useful for anything? And so we developed some tools, basically everything borrowed from neuroscience again, to manipulate the bioelectric states in these tissues. So what we do not do is any kind of electric field application. So there are no fields, there are no electrodes, there are no frequencies or electromagnetics or waves or anything like that. What we do is we manipulate the native interface that cells are using to hack each other, which means we can open and close these gap junctions to control the topology of the network, and we can open and close the various channels and pumps to be able to control the bioelectric state of each cell. And so we use pharmacology, we use optogenetics, all the same kinds of tools that neuroscientists use in the brain and nervous system. And so now comes the main question. Okay, so can we use this to ask what information content do these patterns have? And I showed you a moment ago that there's this little spot here that sets up the eye for the location of the eye for these cells. This is how they remember where to put the eye. So so one thing we were able to do is to take some potassium channel RNA and inject it in a different location of a frog embryo to try to induce that same bioelectric eye-like pre-pattern somewhere else. Well, if you do that somewhere else, those cells get the message and they do exactly the same thing and they build an eye. And you can do this anywhere. You could so here's a bunch of cells that were supposed to be gut that have been given the bioelectric memory that says no, you should be an eye, and that's exactly what they build. And if these eyes, if you section them, you can see lens, retina, optic nerve, all the right components. So there's a few interesting things here. First of all, clearly these bioelectric patterns are instructive. It is not just an epiphenomenon of other things that happen. They actually control what organs get built. That's first. Second, it's incredibly modular. So the eye has a very complex structure, lots of different tissue types. We didn't have to specify any of that. All we said was build an eye here. That's it. Everything else is it's like a simple stimulus that triggers all kinds of complex behaviors down the line. So it's incredibly modular. Another thing is that if you read the developmental biology textbook, they will tell you that only these cells up here are competent to make an eye. Only the anterior neuroectoderm can make an eye. Well, that turns out to be a limitation on our competence, not on the cellular competence. And this is, again, an important lesson in this basal cognition field, is that every time you estimate the competency of a system or the intelligence, you're basically taking an IQ test yourself. All you're saying is, this is what I've been able to figure out. And so if we prompt the system, not with a PAX6 so-called master eye gene, which truly only works up here, but with the more salient bioelectrical pattern, then you can make this eye anywhere. And then the final interesting thing about it is that if you only inject a few cells, so these blue cells are the ones we injected. This is a lens sitting out in the tail of a tadpole somewhere. If you only inject a few, what they do is they recruit their friends. They can tell there's not enough of them to make a whole eye. These cells out here, all these brown cells, we never touched them. They were secondarily recruited by these guys into this project of making an ectopic eye. And we know other collective intelligences that do this, that recruit other members of the group to help handle bigger problems. And this is all the work of Sherry Au and Vaibhav Pai. In my group. And so we use this kind of thing. I don't have time to go into all the biomedical aspects of it because I want to focus on the cognitive side, but we use these sorts of tools to do all sorts of things in regenerative medicine. For example, try to regenerate legs. So frogs, unlike salamanders, normally do not regenerate legs. With the appropriate bioelectrical state, you can convince these cells that they should in fact go towards leg growth and not scarring. And you can see what happens here. Within 45 days, you've already got some toenails, some toes, you've got a toenail. Eventually, a pretty nice leg is regenerated. And again, this is triggered by a very simple stimulus. We do not try to micromanage this process. This is not scaffolds, 3D printing, stem cells, nothing like that. The goal is to convince the cells right at the first day after injury to go down the path of regeneration, not the path of scarring. And at this point, I have to do a disclosure because Morphosceuticals is a company that Dave Kaplan and I founded that is seeking to move all of this stuff using wearable bioreactors to mammalian regeneration and hopefully eventually human patients. So I've made the claim, and I've shown you that these bioelectrical patterns are instructive for what it is that the cells are going to build. And I want to specifically show you an example of reading and writing one very specific kind of pattern memory. And for that, I will introduce you to this creature. This is an amazing kind of model system. These are planaria, flatworms. And if anybody has questions about this, we can talk at the end in the Q&A of what's going on with them. But one of the key things about them is that you can cut them into pieces, and each piece will regenerate a perfect little worm. So each piece has everything it needs to build a perfect little worm and stop. So we wanted to ask a simple question: a piece like this, right? This little middle fragment here. How does it know how many heads it's supposed to have? And for that purpose, we again studied the bioelectric pattern, and we found out that indeed in these fragments there's a voltage gradient that says one head, one tail. And we asked, well, what happens if we change this and we duplicate it? And so here it is, and it's a little messy still. The technology is still being worked out, but you can see here what we've done is we said two heads. And if you do that, the cells will, sure enough, they will build a nice two-headed animal. This is not Photoshop or AI. These are real animals. And the most important thing here is this bioelectric pattern is not a readout of this two-headed animal. This bioelectric pattern is a readout of this animal, which anatomically looks perfectly normal. Anatomically, it looks normal, one head, one tail. Molecular biological markers are normal, so the head marker in the head, not in the tail, just like this. So at the level of anatomy and at the level of gene expression, this guy's completely normal. What's not normal about him is that we, meanwhile, changed his internal representation or memory of what a correct planarian should look like. In fact, I think this is the basis of counterfactuals, the basis of this amazing capacity that brains have for time travel, mental time travel, of remembering and foreseeing things that are not true right now. This bioelectrical state is not true right now. As at the moment of this one-headed animal, this is not correct. It doesn't match. But this is a representation of what it's going to do in the future if it gets injured. If it does not get injured, it simply stays one-headed, and that's that. But if it does get injured, the cells use this information to decide what it is that they should be building, where in anatomical space should they be going, what is the correct pattern, and then they go ahead and they build this, and then they stop. Because this memory, this is their reference point of what a correct planarian should look like. So a standard Planarian body can hold at least one of two different representations of what a normal Planarian should look like, and I think actually many more. But this is a story I'm telling today. So one reason I keep calling it a memory is that it has all the properties of memory. For example, once you've created these two-headed worms, you can keep cutting them in plain water, no more manipulation of any kind, and you will see that they hold state. Once you've convinced those tissues that the right Planarian is two-headed, every fragment will reconstruct that same pattern. The memory is reinforced and duplicated, and they will continue to build two-headed worms in perpetuity. But we do know how to set them back. Now we can force the memory back to normal, and you can get them back to one-headedness. So here you can see these two-headed animals. There's some fascinating behavioral questions to be asked about what it's like to have two brains in one body, and how do they behave and so on. But one other thing to keep in mind is that what this memory really is a navigational aid about where in anatomical space they should go. And that means that we can go beyond just the number of heads to the type of heads, because actually the evolutionary morphospace here contains many different shapes of heads. There are other species that have other different types of heads, and they live in these attractors in this anatomical morphospace. And so now the question is, well, could we get the exact same hardware to visit one of these other anatomical regions? In other words, what would it take to convince these cells to build something completely different? So it turns out that you can do this by, again, manipulating these bioelectrical patterns. So here's a Dugisia doratocephala with a nice triangular head like this. If you amputate that head and you manipulate the bioelectrical pattern that normally guides them to their normal region of anatomical space, you can get them to go somewhere else. They can make flat heads like a P. felina. You can make round heads like an S. mediterranea. Of course, they can make their own normal ones. Not just the shape of the head, but actually the shape of the brain and the distribution of stem cells becomes just like these other species between 100 and 150 million years distance. So let's remember, no genetic changes anywhere here. This is completely stock, out of the box hardware. Their genomes have not been edited. There are no transgenes. So the two-headed form, the different head shapes, all of this is handled at the level of the decision-making of how they're going to navigate this anatomical space, and they're able to build all kinds of things. In fact, you can go much further, and you can visit regions of that space that normally don't belong to Planaria at all. So you can make these crazy spiky forms. You can make cylinders, three-dimensional. These guys aren't even flat. You can make three-dimensional cylinders. You can make some hybrid shapes like this. And so you can imagine exploring this latent space of possibilities, as Darcy Thompson already foresaw in a long time ago. And it's also interesting to note that we aren't human bioengineers aren't the only hackers who do this. Evolution does this all the time. This is a hedgehog gall produced by a wasp, a non-human bioengineer that basically prompts these leaf cells to build something completely different. This is not made by the wasp. This is made by the leaves under a stimulus from the wasp. we would have had no idea that these cells, which are reliably 100% of the time build a nice flat green architecture, we'd have no clue that they're capable of something like this. So this again is reminding us that with the right prompts, many systems are capable of behaviors that we actually have no idea about. And so you know, it took evolution a long time to make a wasp that has the signals to get it to do that. And one wonders, and we are now checking with modern tools of morphoengineering and AI, could we learn to communicate these goals much more efficiently? So in this last little bit, I just want to pull back a little bit and come back to this broader idea of diverse intelligence. So what we've been looking at is the behavior of a weird and unconventional intelligent agent. It's a collective. Instead of neurons bound together by bioelectricity to solve problems in three-dimensional space, which is what all of us do, it uses those exact same mechanisms, so same ion channels, same neurotransmitters, to navigate anatomical space and to solve problems, to have novel behaviors and so on in the anatomical space. So can we come up with a way to think about all of these different diverse agents? What do they all have in common? You know, what is fundamental to being an agent, whether you're artificial or natural or you know, whatever space you're operating in? What is fundamental? And one thing you can think about is I call this the cognitive light cone. This idea that we can map out on let's collapse all of space onto one axis, all of time onto this axis. Let's map out the size. Let's map out the size of the largest goals that the system is capable of pursuing. And so oops. Here we go. So you can imagine that these different creatures have different goals that they pursue. So the set points, the size of the set points, and the size of the things that stress them and spur them towards new behaviors, you know, can be very, very different size, but they can all be put on this continuum. And so basically, what we have in biology here is that the little tiny, very, very low scale goals of single cells, things like pH, hunger level, various physiological parameters at the scale of a single cell, can be scaled up in electrical networks to become these massive construction projects. And so what we're looking at here is how living systems scale their competencies to bigger cognitive light cones, meaning bigger goals in new spaces. So here's the cell. Evolution allows this incredible scale up. So instead of just working on maintaining specific states here at the single cell level, these cells are working towards this massive construction project. Many of them, in fact, will die doing this. It's fine because they're not committed to their own individual set points. They're committed to making this journey in anatomical space as a collective. And this process has a failure mode. That failure mode is cancer. So what can happen is that if these cells disconnect from the collective, they can no longer remember these enormous, grandiose goals. All they can remember are their ancient unicellular goals of reproduce and maintain certain physiological and metabolic parameters. So that's what this is. This is human glioblastoma. And so one way to think about this then is as cancer as a breakdown of collective intelligence. If we could also talk about the parallels between this and various dissociative disorders, meaning breakdowns in the unified consciousness of neuronal groups in human patients. And so this is basically a kind of dissociative disorder of the collective, of the anatomical collective intelligence. And that reminds us that these cancer cells are not more selfish. They just have smaller selves. Right? The goals, the size of the goals they're working on are very much smaller. The boundary between self and world has shrunk. They no longer see the rest of the body as part of themselves. They're just you know, individual amoebas now. And that suggests a completely new approach to cancer. So what you can think about is, instead of trying to kill these cells, so toxic chemotherapy, you could try to simply reconnect them back to the collective. And so that's what we did here. We injected nasty human oncogenes. And this includes P53, KRAS, dominant KRAS mutants, and so on. Normally, they make tumors. But if you co-inject an ion channel that forces these cells to stay in proper electrical communication with their neighbors, meaning you hyperpolarize them, the gap junctions stay open, what happens is that even though the oncoprotein is blazingly expressed, so here it is labeled in fluorescent, in fact, it's all over the place, there's no tumor. This is the same animal. There's no tumor because even though the hardware is damaged, being part of the collective allows the cell to keep working on something much larger. These goals of making nice skin, nice muscle, and so on. And so this is an example of how thinking about very fundamental questions. You know, how do unified minds arise from competent collective, from competent parts, leads you to a kind of a therapeutic roadmap. And we're at this point we're now pursuing this in human cells and hopefully approach to cancer medicine. So the last thing I want to talk about is where do these goals actually come from? So we've talked about homeostasis. We've talked about intelligence as the pursuit of specific goals. We've talked about different size of goals. And I want to, in the last few minutes, just talk about where do these goals come from. And just to remind ourselves a little bit about the scaling of these goals. This is, we normally look at an early embryo and we say, okay, there is one individual. And I want to just remind us that, again, there isn't one of anything. If you look at the blastoderm, there are many, many cells. And in fact, if you make little scratches, and I used to do this as a grad student with duck embryos, if you make scratches, each of these domains, not feeling the other one for about 4 hours until it heals up, will self-organize into an embryo. And this is how you get twins and triplets, in fact, conjoined multiples. And so the question of how many individuals, how many cognitive and functional individuals are in this embryo is not clear. The genetics does not specify. It could be 0, it could be 1, or it could be probably up to about half a dozen, depending on the signaling that goes on and depending on where these systems set the boundaries between self and outside world. Because every embryo, every cell has some other cell for its neighbor. So where does one embryo end and the other embryo begin? And so there's some fascinating issues here, again, about individuality, self-organization of minds as well as bodies. And I want to mention briefly two mechanisms by which this collectivity happens, because actually what you need to have here is alignment, right? What the reason you call all of this one embryo is because all of these cells are in complete agreement about where in anatomical space they are going. They are aligned both physically. I mean, they literally get aligned in planar cell polarity, but in terms of their navigation, they are all in agreement about where they're going. And so there are two mechanisms that we've studied that allows that to happen. One is anonymization of memories. So when under normal signaling, if this cell sends some kind of a signal to that cell, it's very easy for this one to know that signal comes from the outside. They can attend to it, they can ignore it, whatever. It's very easy for them to maintain separate memories and separate goals and so on. But these gap junctions are kind of special. What happens when you form electrical synapses is that there is a direct conduit from the internal milieu of one cell to the other. And that means that it becomes very hard for them to have separate memories. When something happens to this cell, and let's say it generates some kind of calcium spike as a signature of the memory of that event, that will propagate to this cell. It doesn't have any metadata on it about who it belongs to. This cell receives that information. And at this point, it has no idea, was that my memory? Was I the one that got poked and have this calcium spike, or was that somebody else? And so they have kind of a mind meld in the sense that information passes. And so now while this was a false memory according to this cell, it is actually a true memory according to the new collective that is formed here. So sharing memories and a difficulty of keeping individual histories and individual goals is part of what happens when you bind groups of cells, which used to be individual animals, into collections that have a aligned common goal. So that's one kind of mechanism. Another kind of mechanism, which is not particularly bioelectric, it is stress sharing. So what happens here is that imagine, and this happens in all kinds of spaces, but I'll just talk about physical space here. Imagine that this cell wants to get up here. It's in the wrong location. And it's very stressed out because of it. I mean, cells literally have stress markers that go up when their goals aren't met. And if it were just to stay that way, these other cells would never cooperate with it and let it go because they're very happy. So their energy is at the lowest level. They're perfectly happy where they are. They're not moving. But what happens is that these cells can now export their stress signal. They leak. They leak the molecules that indicate stress. Well, when that happens, these other cells start to experience it. Again, much like with what I just showed you, these molecules are all the same. They're all conserved. And so when these cells have it, they are now also stressed. The plasticity goes up. They start to move around because they're no longer sure they're in the right place. and then the cell can do what it needs to do. Put another way, the reason to export stress is that it increases collectivity, because your problems, my problems become your problems, right? By sharing stress, everybody is now motivated without any kind of additional mechanism for altruism. You don't need to be altruistic. You just need to experience some degree of the same stress that I feel in order for the plasticity to go up, and everybody rearranges and gets to where they're going. And we've done all sorts of computational modeling of this. Okay, so the very last bit here is this. We've seen new ways to look for intelligent problem solving in other embodiments. We've looked at new ways to use the bioelectrical interface to communicate, not micromanage, to communicate with this morphogenetic intelligence and get large scale outcomes that would be very hard to try to create bottom up. And we've looked at a couple of policies for the alignment of parts that I think are part of this cognitive loop mechanism. So the final thing, where do these goals come from? Well, typically, when we look at any animal or a plant or anything like that, and we ask where do its behavioral and structural properties come from, you say, well, evolution, eons of selection. So everything that wasn't that way died out, and so now here it is. So in order to test this, we created some new synthetic living materials that do not have a history of selection in this particular embodiment. So here's one example, and so this is the work on xenobots by Doug Blackiston, where we take some epithelial cells from an early frog embryo, we set them aside, and they could do many things. They could die, they could spread out and walk away from each other, they could form a two-dimensional monolayer like cell culture. What they do instead is they self-assemble. Here, this is a time lapse. They self-assemble into this kind of a spherical thing. The flashes you're seeing are calcium signaling. And remember, there are this is just skin. Okay, this whole thing is just epithelial cells. And then they do something interesting. They use the cilia that they have on the outside. The cilia is normally used to redistribute mucus down the body of the frog. And what they use these cilia for, they start to row against the water and they swim. So they can swim back and forth like this. They can go in circles. They have certain collective behaviors where they can interact. They can sit quietly. This one's going on sort of a much longer journey. Here is one traversing a maze. So what you will see is it swims down here. It takes the corner without having to bump into the opposite wall. And then at this point, spontaneously, it turns around and goes back where it comes from. We don't know why, but you can imagine trying to track the calcium activity during this process to see what is happening when it decides to turn around and then go back. So they have these autonomous motile behaviors. One of the most amazing things they do is they make copies of themselves. Now, how do they do that? We've made it impossible for them to reproduce in the normal froggy fashion. They don't have any of those organs, but turns out that if you supply them with loose skin cells, so that's what this white stuff is, they're just loose epithelial cells, they will run around and collect them into little balls and polish these little balls, both individually and as a group here. And because they're working with these cells, which are also an agential material, not just passive particles, these little balls mature into the next generation of xenobots. And guess what they do? They run around and they do exactly the same thing, making the next generation and the next. So we call this kinematic replication. So what did the frog genome learn over its time on Earth? Well, it certainly learned to do this. This is what you see if you just look at normal development. They do this developmental sequence, and then you get these tadpoles that swim around. But turns out they can also do this. And what's interesting here is that we did not touch the genome. We didn't add any scaffolds, no synthetic biology circuits, nothing. All we did was to liberate these skin cells from the influence of the rest of the cells. If you take them away from the rest of the cells, you get to find out what they normally do. Under what they want to do under normal conditions, they're basically bullied by these other cells to be like this boring two-dimensional layer that keeps out the bacteria. But they can have a much more interesting life if you get them away from all of this. This is their developmental sequence. This is an 84-day-old xenobot that Doug made. We have no idea what it's turning into or what's going on. Where does this developmental program come from? And then they have completely different behaviors like kinematic self-replication. So where is all this coming from? They don't have a history of being selected to be a good xenobot. There's never been any xenobots. We are currently studying their behavioral capacities. Stay tuned for that. That stuff will come out hopefully later this year. So I'm not making any claims about that right now, except to say that whatever features they have, a history of selection is not a good answer here. So we need to understand. And to go further than that, I mean, you might think, well, frogs are pretty plastic, amphibian embryos are plastic. So I'll show you this, and this is the work of Gizem Gamushka, a PhD student in my group. And this is, if I ask you what you think this is, it looks like some sort of a primitive creature that you got from the bottom of a pond somewhere. If you were to sequence the genome of these cells, what you would see is 100% Homo sapiens. These are adult human patient cells. They come from the trachea. They self-assemble into something we call an anthrobot. Again, much like the xenobots, it has its own shape. It has its own behavior that is nothing like what these cells do in vivo. Excuse me. And so then we might ask, well, we know what tracheal epithelia do in the patient. What do these things know how to do, and where would where would it come from? So we're only now beginning to scratch the surface. One thing they can do, if you put them on a dish of human neurons, which have been scratched. So here's a wound. Here's a big scratch wound. What they will do is assemble into a superbot like this, and heal and try to get the two ends and try to connect them together. So 4 days later, you pick it up and you see under where the bot was sitting, this is what it did. It took the two sides and healed them together. Now, who would have thought that these tracheal cells, which sit there quietly in a sheet in your airway for decades, that given the opportunity to reboot their life into a new form, they would have all these capacities. And we've been studying them. They have thousands of new genes they express that are different from what human tissue expresses normally, and so on. So I'm gonna just end here by saying that, look, I think that everything that Darwin was looking at when he said endless forms most beautiful, so all the natural examples of what the material can do, is like one tiny corner of this enormous option space of new bodies and new minds. All kinds of combinations of evolved material, engineered material, and software is some kind of agent. So cyborgs, hybrids, biobots, chimeras, all of these things are being made. We will be living with many of them in the decades to come, and we must understand how to ethically have a kind of a symbiosis with them. This word actually was discovered by GPT. I asked it for for a word that kind of gets this idea across that we need to be in a kind of ethical symbiosis with these synthetic life forms. So I really think we need platforms to better understand what it means to interact with other intelligences in other embodiments. So what I've tried to argue today is that intelligence goes beyond brainy organisms in 3D space. This occurs in many spaces that are hard for us to visualize. That cognitive glue that binds these pieces together is a really attractive target for biomedicine, in particular bioelectricity, and shows us that neuroscience is not just about neurons. Many of the tools and concepts, everything from active inference and perceptual multi-stability and memory and all learning, all of these things can be repurposed for some amazing applications in biomedicine and robotics and so on. But the broader issue here, I think, is that we really need to develop the field of diverse intelligence. This is telling us that one of the ways that I think we need to mature is to get better at discovering and relating to these unconventional intelligent agents. So if you're interested in more details, these are some of the papers that dive into all of this stuff. And so I need to thank all the people who did the work. So this is Doug Blackiston, who did all of the biology for the Xenobot models. Vaibhav Pai and Sherry Au did the bioelectric eye induction. Brooke Chernet is cancer and the bioelectric control of cancer. Narosha Murugan did some great work on the frog leg regeneration project. Fallon Durant did the pattern memories, the latent pattern memories in Planaria, and Gizem Gamushka is the anthrobots. So I'll thank all of these people. Our many collaborators, all the other folks in the lab, our funders who have been supporting this work. Jeremy did all the illustrations, and here are the disclosures. These are 3 companies that have supported our work. And as always, all the heavy lifting is done by the animal model systems. So thank you very much, and I will stop there.